Welcome to another episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor. Welcome to another episode of our show, bringing another really awesome guest today, involved in science and technology and biomedicine, theoretical gerontology that is creating a better tomorrow for all of us. Today we are headed out on our virtual road trip, as I like to say, and we're headed over to China to meet with a fascinating young man who I and many others, I believe initially bumped into in the social media spheres by the name of Bilu Huang, who is a visionary that is dedicated to finding solutions to some of the most pressing challenges facing humanity with an initial interest in biological aging. And he has been working as an independent researcher whose, fascinatingly enough, knowledge has been entirely self-taught. And he has been spending a lot of time working in the trenches of theoretical gerontology, creating novel concepts of why our cells age. He has proposed a really interesting model known as the Telomere DNA and Ribosomal DNA co-regulation model for cellular senescence, which he's been working on for several years, experimentally validating, and using these theories has also recently gotten into the entrepreneurial space, setting up a biotech company by the name of Fuzhuang Therapeutics to ultimately further develop and translate these concepts of rejuvenation of cells and ultimately thinking about, curing various age-related degenerative diseases. A really cool guy, a lot of interesting themes to get into today, and honored to have him with us. Bilu Huang, thank you so much for taking the time to come on the show today.
Hi Ira, thanks for having me on the show. I am honored to share my telomere DNA and ribosomal DNA co-regulation model for cell senescence TRCS with you and your audience.
Bilu, I'd like to start out just by handing you the floor for a little bit to further introduce yourself and a bit about your background story. I know a lot of us have been following you in social media, a lot of your information that you've been putting out there on Twitter, but take a little time to introduce yourself, where you grew up, where you went to school, and ultimately what first got you interested in the general area of science and technology. And then we'll head further into the area of aging.
Hello everyone, my name is Bilu Huang. I was born in 1972 in a common family in Xiaqiao Village, Xinyang Town, Yuxi County, Sanming City, Fujian Province, China. As far as I can remember, there were no highways or electricity in my hometown. I only attended Xiaqiao Primary School. After graduating from primary school, I dropped out and started working. I learned about biology and other subjects entirely on my own. I first became interested in science and technology when I was 11 years old. One time, I flipped through a chemistry book that belonged to my brother. I saw pictures and descriptions of atoms and molecules. For the first time, I learned that visible matter is made up of very tiny molecules, which are composed of atoms. Atoms consist of a nucleus and electrons that rotate at high speed around the nucleus. The nucleus is made up of neutrons and protons. Matter may have infinite divisibility. This made me very interested in chemistry.
Well, tell us now a little bit about what got you interested in studying the area of human aging.
When I was 8 years old, one day I went with my father to a vegetable field not far from our home. The field was divided by a deep gully eroded by water. I saw a niche in the gully wall, inside which was an earthenware jar covered with green moss. I asked my father what was inside the jar. He said that people age and die, and then they decay until only a pile of white bones remains. The jar was for holding the bones of the deceased. I suddenly felt a chill run through me. When I touched my forehead, I felt a sense of desolation. That night, I had a hard time falling asleep. I kept thinking in bed, one day I will also age and die. Death means the disappearance of consciousness, and the remaining bones will be placed in a dark jar. In the universe, space and time are infinite, yet each of us can only live a short life once. Once dead, we will never come back to life. The more I thought about it, the more scared I became. So, I made up my mind to conquer aging and to understand the nature of consciousness. After graduating from primary school, I began to observe the aging phenomena in animals and plants, and collect various theories about aging.
One of the things that I've seen you talk quite a bit about in the social media is the history, let's say, of a lot of the theories that have been proposed over the years for why humans age, many which, as you pointed out, have not really yielded any success, many which have been disproven over the years, from free radical theories to rate of living theories and so forth. Take a little bit and sort of talk a little bit about this history from your own perspective.
Many of our degenerative diseases are caused by aging, and cancer is also a result of the aging of the immune system. Therefore, understanding the mechanisms of aging is of great significance. To this end, humans have proposed a wide variety of aging theories. In 1990, Medvedev and others counted more than 300 published aging theories, and each theory could potentially lead to corresponding anti-aging methods. The most reliable way to test whether an anti-aging method is effective is to see if it can extend the lifespan of wild-type mice. Unfortunately, according to the annual analysis of the effects of lifespan-extending drugs and compounds by drug age, although there have been more related studies in recent years, no more significant lifespan-extending effects have been shown. Even many so-called anti-aging drugs that claim to extend lifespan cannot pass the rigorous ITP, Interventions Testing Program tests conducted by the U.S. National Institute on Aging. This suggests that these aging theories may be incorrect. If the theory is wrong, all the efforts are in vain. For example, in 1956, Harman proposed the free radical theory of aging, which suggests that reactive oxygen species produced by mitochondria attack various macromolecules, causing a variety of damages. However, the free radical theory of aging has been debunked. Methylene blue, MB, is a powerful antioxidant that can penetrate into organelles such as lysosomes and mitochondria. The ITP test found that methylene blue did not extend the average lifespan of mice. Alpha-lipoic acid is an antioxidant that is good at preventing lipid peroxidation to stop the accumulation of its harmful byproducts, including harmful aldehydes such as acrolein. However, alpha-lipoic acid significantly shortened the median lifespan of mice. In 1980, Mikel and others proposed the mitochondrial theory of cellular aging. This theory suggests that reactive oxygen species produced by mitochondria can cause mutations in mitochondrial DNA within cells. As age increases, the amount of mutated DNA accumulates, leading to mitochondrial dysfunction and insufficient cellular energy. which in turn causes cells to enter a state of aging. In fact, the mitochondrial theory of aging had already been debunked before it was proposed. In 1958, Yoshida found that within 8 hours, the chloroplasts in the protoplasts with nuclei of Elodea densa underwent a process of aging and structural destruction, while the chloroplasts in the protoplasts without nuclei remained green and continued to accumulate starch. In 1975, Wright and Hayflick transplanted young nuclei into enucleated senescent cytoplasm, and the cells regained youthfulness and continued to divide according to the remaining number of divisions of the young nuclei. Conversely, when senescent nuclei were transplanted into enucleated young cytoplasm, the cells exhibited senescent phenotypes, and their division numbers were significantly reduced. Both studies indicate that the determinant of cellular aging is not the mitochondria, but the cell nucleus. Moreover, Vermost and others found that mouse models with increased mitochondrial DNA mutations showed no signs of accelerated aging and did not have shortened lifespans. Many other theories of aging have also been debunked one after another. For example, the inflammation theory of aging. This is because the anti-aging drug NR, which can boost NAD+, can reduce the release of inflammatory factors by 52.6%, and the macrophage migration inhibitory factor antagonist MIF098 can help macrophages move and reduce chronic inflammation in the body. However, after the most authoritative ITP test by the NIA, it was found that NR actually shortened the lifespan of male mice by 3%, and MIF098 could not extend the lifespan of mice. Studies on the Tisimeni people in the Bolivian Amazon rainforest and the Orang Asli people in Malaysia have also found that there is no obvious correlation between the inflammatory aging score and hypertension, diabetes, or even chronic kidney disease. The theory that protein misfolding causes cellular aging has also been debunked. This is because teprenone GGA induces the expression of heat shock proteins in mammalian tissues, ensuring that proteins fold correctly. However, after ITP testing, it was found that teprenone does not extend the lifespan of mice. DNA damage has long been considered a possible major cause of cellular aging. However, this theory has already been debunked. Ionizing radiation can greatly increase damage to both nuclear DNA and mitochondrial DNA. But sublethal doses of ionizing radiation can actually extend the lifespan of fruit flies, houseflies, rats, and mice. In Japan, atomic bomb survivors have longer lifespans and fewer cases of cancer than the average population. It is generally believed that the stronger the DNA repair capacity, the longer the lifespan. However, cockroaches and tardigrades have extremely strong DNA repair abilities, yet under suitable conditions. Their lifespans are only a few months. The autophagy theory of aging suggests that enhancing cellular autophagy can accelerate the degradation of toxic waste molecules in cells, such as mutated mitochondria and denatured proteins, thereby exerting an anti-aging effect. However, paradoxically, it has been found that ovarian aging in mice is associated with enhanced autophagy in granulosa cells. Chloroquine is the only autophagy inhibitor approved by the FDA. Yet, chloroquine can extend the maximum lifespan of rats by 13%, which is comparable to some of the best anti-aging drugs ever tested in mouse models, inhibiting autophagy in aged C. elegans, a type of nematode, can extend their lifespan by 50%. Conversely, increasing autophagy in the intestines of C. elegans can actually accelerate aging. The epigenetic theory of aging posits that the problem of aging lies in epigenetic drift. This includes the much-hyped information theory of aging, proposed in recent years by David Sinclair and others. However, in 2016, during the first attempt to use Yamanaka factors to partially reprogram the epigenome, it was found that this approach did not extend the lifespan of wild-type mice. By 2024, Altos Labs, a company invested in by one of the world's richest men, Jeff Bezos, to the tune of $3 billion, had managed to increase the median lifespan of wild-type mice by only 12% through partial epigenetic reprogramming. This result is not as impressive as that achieved by small molecule anti-aging drugs. Other studies have found that, during partial epigenetic reprogramming, although the cellular identity remains unchanged, telomeres do not lengthen or may even shorten slightly. Once the expression of Yamanaka factors is halted, signs of aging accumulate rapidly again, including a swift return of the epigenetic age to its pre-reprogramming state. This indicates that the effects of partial epigenetic reprogramming by Yamanaka factors on aging intervention are transient. This suggests that the approach of intervening in aging through partial epigenetic reprogramming has already failed. As early as 2011, a group of scientists in the United States gave wild-type aged mice a drug that could clear senescent cells from many tissues in the body. The result was that the lifespan of the mice did not increase. At that time, Professor Fu Wenqing from Fujian Normal University and I analyzed that clearing senescent cells might accelerate aging. This is because the removal of senescent cells stimulates surrounding cells to replicate and differentiate to fill the gaps, which in turn promotes replicative senescence in cells. Therefore, the anti-aging intervention of clearing senescent cells may be wrong. Recently, it was reported that Unity Biotechnology's stock price has plummeted and the company is deserted. The reason is that the Phase 2 clinical data released by Unity showed that the drug UBX0101, which precisely clears senescent cells in the joints, did not show any statistically significant difference compared with the placebo in treating knee osteoarthritis pain. Previous studies have also shown that female mice that started taking dasatinib and quercetin to clear senescent cells from a young age actually aged faster. The treatment of ABT263, which clears senescent cells, accelerates ovarian aging in aged female mice when dasatinib and quercetin are used to clear human senescent cells. A significant increase in epigenetic age and a significant shortening of telomere length are observed. Therefore, the once highly anticipated anti-aging intervention of clearing senescent cells has also failed. Finally, I would like to talk about the much-hyped anti-aging method of injecting young plasma in recent years. The idea of vampire therapy has existed since ancient times. But does young blood really have the effect of rejuvenation? Obviously not. This is because laboratories use calf serum and fetal bovine serum to culture cells every day. Yet cells still cannot escape the fate of aging and death. In 2014, a research paper reported that old mice were repeatedly injected with young plasma and compared with the control group that received saline injections. The median lifespan of the control group was 27 months, while that of the plasma treatment group was 26.4 months. Not only did it fail to extend the lifespan, but it also shortened it a little. Therefore, the anti-aging intervention of injecting young plasma has also failed. In addition, resveratrol, metformin, NR, which boosts NAD+, curcumin, and other drugs that claim to be effective in anti-aging have all failed the ITP test. In 2023, a review titled Hallmarks of Aging, an expanding universe was published in the journal Cell. It listed the 12 major hallmarks of aging. However, I believe that the view of using these hallmarks as intervention targets for aging is wrong. This is because the hallmarks of aging are not the causes of aging, but the results of cellular aging. Therefore, interventions targeting the results of aging will be of little effect, and only address the symptoms, not the root cause. To avoid misleading people with wrong ideas, I recently published a paper titled Causality of Aging Hallmarks in the journal Aging and Disease. In this paper, I unified various hallmarks of aging as the results, mediated by the tumor suppressor protein p53 through telomeres and rDNA. You are welcome to have a look.
Another thing that you talk about, and I sort of sit back and I enjoy, well, maybe not enjoy, but I watch the ongoing sort of fiercely debated core issue in aging research between programmatic theories and damage accumulation theories. And I don't think that this debate has ever subsided. I'd like you to take a little bit of time, talk about your perspective from both these areas, the current problems associated with both of them, and sort of the current problems with regard to how research is being approached in relation to them.
In the process of exploring why humans age, I learned that theories of aging can be broadly divided into two major categories: the damage accumulation theory and the program theory. The program theory posits that both the development and aging of an individual are controlled by preset programs. In contrast, the damage accumulation theory suggests that aging stems from random damage to molecules and cells, which accumulates over time and cannot be fully offset by the repair systems. Theories such as the free radical theory and the theory of entropy increase fall under the category of damage accumulation theories. This framework encompasses damage to nuclear and mitochondrial DNA, misfolding or cross-linking of proteins, and the accumulation of metabolic waste products like lipofuscin. Later, I came across some literature indicating that the 5 kilobase deletion in mitochondrial DNA in the human brain does not occur in children. The 3.4 kilobase deletion in mitochondrial DNA in the human diaphragm increases with age, but is zero in individuals under 30. Ionizing radiation can cause damage to both nuclear and mitochondrial DNA, as well as cross-linking and denaturation of proteins. However, sublethal doses of radiation have been shown to extend the lifespan of fruit flies, houseflies, mice, and rats. Feeding mice with antioxidants has been found to reduce the content of lipofuscin in cardiomyocytes and neurons, but this did not lead to an extension of the mice's lifespan. These pieces of evidence all suggest that the accumulation of various types of damage is not the cause of aging, and that such damage can be overcome. The reason these damages accumulate with age is that programmed aging downregulates these defense mechanisms, causing the repair rate to fall behind the rate of damage. Based on this, I discarded the damage accumulation theory of aging and shifted my focus to the program theory of aging. Infants begin to grow milk teeth at around 6 months after birth, start to replace them with permanent teeth at the age of 6, and typically begin to grow facial hair around the age of 16. At around 40, presbyopia, age-related farsightedness, often sets in, and so on. Silkworms hatch from eggs into tiny larvae, undergo several molts, then transform into pupae, and finally emerge as moths. After mating and laying eggs, they die. Moreover, each species has a relatively fixed timetable for growth, development, maturation, aging, and death. Therefore, the essence of individual development, maturation, aging, and death is a program. If the program theory of aging is correct, then the process of cellular aging must involve the programmed expression of gene clusters along the timeline on the chromosomes. The process of aging and death is essentially the gradual shift from a beneficial gene expression profile to a detrimental one. This is indeed the case. For example, hepatocytes primarily express alpha-fetoprotein during the embryonic stage, albumin after birth, and as they age, the albumin gene is gradually turned off in favor of expressing other proteins. Since albumin is an indispensable protein, measuring albumin levels can predict a person's proximity to death. Young macrophages mainly express the isoform VEGFA165A of vascular endothelial growth factor A, which promotes angiogenesis and thus enhances tissue repair. In contrast, aged macrophages predominantly express the isoform VEGFA165B, which inhibits angiogenesis and thereby affects tissue repair. The scavenger receptor SRB1 is upregulated in aged vascular endothelial cells, actively increasing the uptake of low-density lipoprotein cholesterol, LDLC, from the blood, thereby promoting the progression of atherosclerosis. Clusterin CLU is upregulated in aged hematopoietic stem cells, driving their differentiation towards the myeloid lineage and increasing the production of myeloid cells. During the aging process of hematopoietic stem cells, approximately 1,500 genes are downregulated, and 1,500 genes are upregulated. This is mainly characterized by the upregulation of stress response and inflammatory genes that are harmful to health with increasing age, while the downregulation of chromatin remodeling and DNA repair genes that are beneficial to health. Based on this, the common anti-aging approach is to inhibit an upregulated gene and activate or overexpress a downregulated gene. However, such interventions at the metabolic and signaling pathway levels can only slightly extend lifespan and come with significant side effects. They certainly cannot reverse aging. This is why, despite the discovery of more anti-aging drugs and compounds in recent years, there has been no more significant extension of lifespan. Whether the aging process is controlled by a program or the result of the accumulation of random damage is a fiercely debated question in aging research. Both sides agree that the rate of growth and development and the timing of sexual maturity are controlled by programs. However, after sexual maturity, those who oppose the idea that aging is programmed argue that aging has a negative effect on individuals. Since natural selection can only act on individuals, it is not possible for individuals to develop and maintain a program that is disadvantageous to themselves. Therefore, aging can only be the result of the gradual accumulation of damage that occurs randomly. Given that life, through its long evolution, has developed redundant repair systems to overcome various types of damage, I believe that even after sexual maturity, aging cannot be the result of the gradual accumulation of random damage. Aging after sexual maturity is still controlled by a program. Take the short-lived killifish or other species living in Africa as an example to illustrate that aging after sexual maturity is still programmed. The eggs laid by killifish will go into dormancy during the dry season and hatch again when the rainy season arrives and forms puddles. In Zimbabwe, where there is only a brief rainy season, and puddles dry up quickly afterward, the local killifish strain has a lifespan of only 3 months, equivalent to the length of the rainy season. In Mozambique, where the rainy season is 4 times longer than that in Zimbabwe, the local killifish strain can live up to 9 months. In Tanzania, where there are 2 rainy seasons, the killifish strain can live as long as 16 months. When these 3 types of killifish are raised, under the same conditions in captivity, the differences in their lifespans still persist. This indicates that aging is controlled by a program. because random damage accumulation cannot explain why these three killifish, which are extremely similar in body structure and belong to the same genus, have such vastly different lifespans. That just happened to match the length of the rainy seasons.
It's interesting. You make a very interesting point in your work that in any proper search for a theory of everything, as you quote, a timing substance of some type needs to be present and incorporated to drive the process of aging, and that such a timer must be, as you say, something stable and something that has, in essence, no half-life because humans take a long time to age. I'd like you to take a little while and spend some time on this. I think it's quite important. And as you've pointed out in your work, proteins, RNA, mitochondrial DNA, chemical modifications, nuclear DNA, epigenetic modifications, histones are all things that have been focused on, but they are all quite biologically unstable and don't really fit the full purpose of a timing substance that moves throughout life. Take a little time to break this concept down because I think it's very important in terms of where we're going with your theory.
Within a single lifespan, the sequences and copy numbers of most genes on the chromosomes remain constant. Therefore, to enable the programmed expression of these unchanging genes, a timing substance is required to drive the process. This timing substance is akin to the sand in an hourglass, with a limited quantity that determines the number of cell divisions and the lifespan of a species. In other words, the fundamental cause of cellular aging lies in the timing substance. Since individual lifespans can exceed a century, for example, the Greenland shark can live up to 400 years. The candidate timing substance that drives the genetic program must be extremely stable, without a half-life. Proteins, RNA, mitochondrial DNA, as well as the chemical modifications of nuclear DNA and histones, are all unstable, with half-lives, and exist in a dynamic equilibrium of continuous degradation and replenishment. For instance, DNA methylation and demethylation, as well as histone acetylation and deacetylation, occur simultaneously. Therefore, they cannot form a temporal measure, and do not possess the properties of a timing substance. This means that the fundamental cause of aging does not lie in RNA, proteins, mitochondrial DNA, or various epigenetic modifications. For example, Liesenfelder et al. Experimentally reversed age-related DNA methylation changes, but this effect only lasted for 15 to 100 days. Thus, the information theory of aging proposed by David Sinclair and others is an incorrect theory of aging. Epigenetic age-related changes cannot be the cause of aging, just as wrinkles on the face are not the cause of skin aging, but the result of aging skin cells. Therefore, epigenetic clocks can only be calibrated by more stable reference clocks, such as telomeres and rDNA through mediators like p53. This is indeed the case, as it has been reported that telomeres can influence gene expression by altering the epigenetic landscape. Additionally, rDNA arrays of different lengths have different distributions of heterochromatin and euchromatin, thereby changing the gene expression profile. Since DNA on the chromosomes in the cell nucleus is an extremely stable substance, the timing substance must be hidden within the DNA of the chromosomes. Given that telomeres are a multi-copy tandem repeat DNA array, and their copy number decreases with cell division, telomeres are thus considered the most ideal timing substance. Accordingly, I believe that telomere shortening is one of the fundamental causes of cellular aging. Based on this, in 1995, I wrote a one-page paper and had a friend take it to a biology professor at Tsinghua University. My friend told me that the professor thought my viewpoint had some merit. The content of the paper was roughly as follows. Since living organisms have learned over the course of long evolution how to overcome the accumulation of various types of damage, and given that each species has a relatively fixed timetable for growth, development, maturation, aging, and death, the essence of individual development. Maturation, aging, and death is a genetic program because within a single lifespan, the sequences and copy numbers of most genes on the chromosomes remain constant. A timing substance is needed to drive the programmed expression of these unchanging genes, just as a computer program requires a clock to drive its operation. Based on this, in 1998, I published a monograph titled The Mechanism, Significance, and Treatment of Aging in the Yanjing Medical Communication. The profound understanding of aging presented in the book remains correct to this day. For example, at that time, I already believed that individual aging is caused by the aging of stem cells in tissues, and that the aging of stem cells is due to the reduction in the number of repetitive DNA copies, such as telomeres on the chromosomes.
So, Bilu, at this time, I want you to now take as much time as you want and walk us through what you propose in your core model, known as telomere DNA and ribosomal DNA co-regulation model for cell senescence. And again, take your time with this.
In 1998, Egan et al. Found that corneal endothelial cells maintain long telomeres throughout life. When they studied the telomere length in corneal endothelial cells of people of different ages, the limitation of their replicative capacity is not due to telomere shortening but other reasons. Also in 1998, Bodnar et al. Transfected retinal pigment epithelial cells or fibroblasts with the human telomerase reverse transcriptase HTERT gene, and the cells could only increase their doublings by about 20 times in a limited way. In 2015, John Ramunas et al. From Stanford University delivered HTERT-mRNA to human fibroblasts and myoblasts to extend telomeres instantaneously. Even with multiple telomere extensions, the cells would eventually age. Given that telomeres in non-dividing cells are not easily shortened, there must be another multi-copy tandem repeat DNA array that shortens in non-dividing cells to control their aging. Therefore, I believe that telomeres are not the only timing substance limiting the number of cell divisions and causing cell aging. In addition to telomeres, there should be another timing substance in cells that controls the number of cell divisions and cell aging. Since ribosomal DNA, rDNA, is also a multi-copy tandem repeat DNA array, and its copy number can decrease in non-dividing cells. For example, mature fruit fly somatic cells do not divide, but in their 40-day lifespan, the rDNA array can shorten by half. Based on this, I published a paper titled Telomere DNA and Ribosomal DNA Co-Regulation Model for Cell Senescence TRCS in a Chinese journal in 2021. This theory holds that the shortening of telomeres and or rDNA arrays can lead to an increase in the level of the tumor suppressor protein p53, thereby causing cell aging. In the left is that chromosomes with long arrays of telomeres and rDNA. p53 is rapidly degraded, p53 levels are low, and the cell is youthful. In the right is that chromosomes with short arrays of telomeres and rDNA. p53 is slowly degraded, p53 levels are high, and the cell is aged. Since telomeres and rDNA are universal among all eukaryotic organisms, from yeast and nematodes to mice and humans, the mechanisms of aging across various species have been unified. So, how do telomeres and rDNA drive the operation of the genetic program? Or how do gene clusters on the chromosomes achieve temporal expression along the timeline? Approximately 1/10 of human gene promoters contain p53 binding sites and can thus be classified as p53 responsive genes. p53 not only downregulates the overall rate of protein synthesis, but also acts as a transcription factor that can simultaneously inhibit and activate numerous genes. Therefore, I believe that as age increases, the gradual shortening of telomeres and/or rDNA arrays creates a concentration gradient of p53 along the timeline. Since p53 can bind to the promoters and enhancers of many genes, this results in the upregulation of some genes and the downregulation of others, thereby driving the programmed expression of gene clusters on the chromosomes. Because cells of different differentiation types have differently programmed genetic programs, there are distinct gene expression patterns during the aging process. Whether a theory is correct depends on whether it is self-consistent. If the shortening of telomere DNA and rDNA arrays is the fundamental cause of cellular aging and the timing substance, that drives the programmed expression of gene clusters. Then the telomere DNA and rDNA that are consumed in somatic cells must be replenished in germ cells or early embryonic cells. Otherwise, life would not be able to continue from generation to generation. Fortunately, there is already evidence that the telomere DNA and rDNA consumed in somatic cells can be replenished in early embryonic cells or germ cells. In mice, hematopoietic stem cells experience shortening of the rDNA array due to the activation of mTOR1. The anti-aging drug rapamycin can slow down replicative aging and extend the lifespan of mice by inhibiting mTOR1, thereby inhibiting rDNA transcription and cell replication. Therefore, from first principles, the lifespan of a species is determined by the shortening rate of the telomere DNA array and/or the rDNA array. The telomere DNA and ribosomal DNA co-regulation model for cell senescence, TRCS, that I published in 2021 previously pointed out that the shortening of telomeres and/or rDNA arrays leads to an increase in the level of the tumor suppressor protein p53, causing cells to enter a state of senescence. Moreover, the mechanism of rejuvenation through pluripotency reprogramming is due to the significant elongation of telomeres and rDNA arrays. Accordingly, we knocked down the copy number of 45S rDNA in primary mouse and human cells. The results showed that the senescence markers p53, p21, p16, and SA beta gal were all significantly upregulated, as expected, while telomere length, cell viability, and the number of cell passages were all significantly reduced. We also examined senescent cells in mice, and human embryonic stem cells, HESCS, and human-induced pluripotent stem cells, HIPSCS, and found that both telomere length and the copy number of 45 srDNA were significantly reduced in senescent cells, whereas they were significantly increased in human embryonic stem cells and human-induced pluripotent stem cells. These data strongly demonstrate that the rejuvenation mechanism of HESCS and HIPSCS is not due to epigenetic reprogramming, but rather due to the significant increase in the length of both telomere DNA arrays and 45 srDNA arrays. The fundamental causes of cellular senescence and the Hayflick limit are co-regulated by telomeres and 45 srDNA, with rDNA having a greater weight in the aging process than telomeres. Having introduced why cells age, I will now explain why individuals age. To put it simply, the various organs that make up an individual consist of functional cells and adult stem cells. Functional cells continuously die for various reasons and are then replenished by new functional cells and adult stem cells, generated through the self-replication and differentiation of adult stem cells. However, each time adult stem cells divide, they become a little older than before. The functional cells that are replenished through the differentiation of these aging adult stem cells are also aged, which in turn leads to the gradual aging of organs and the individual. In summary, the aging process is controlled by the genetic program, not the result of the accumulation of random damage. To reverse individual aging and significantly extend lifespan, as well as to achieve organ replacement and cure degenerative diseases, the only solution is to increase the length of telomeres and rDNA arrays in adult stem cells within tissues. This can rejuvenate tissues, organs, and individuals. Apart from this, other anti-aging measures can at best slightly extend lifespan, and are certainly not capable of reversing aging.
So, as mentioned in your bio, you are also, in addition to a scientific thinker and thought leader, you are also, an entrepreneur. You have set up a startup company called Fuzhuang Therapeutics in China to further develop your theories and opportunities based on them. Tell us a little bit about your plans for the future of Fuzhuang Therapeutics.
My next goal is to more widely promote my unique insights into the mechanisms of aging, so that more like-minded people, scientists and bioengineers from all over the world, and more funding can use the TRCS model to guide and accelerate the development of drugs that rejuvenate aging stem cells. This will completely cure various age-related degenerative diseases, replace organ transplantation, and significantly extend human lifespan. I am an independent scientist without an income, and am unable to apply for research funding in China. Therefore, I am currently seeking research sponsorship to further verify my theory and investigate methods of increasing rDNA.
And one other thing I wanted to ask while we have you here today, you're in China. Obviously, there's a lot of interest in aging, longevity, biosciences all over the world. Talk a little bit about the current aging, longevity R&D field, specifically as it pertains to where you are, what's going on in China. Is the interest similar in dynamics to how things are in the United States? Is there a lot of funding flowing into it? Take us a little into the story of what's happening in China.
It seems that China has relatively little interest in the aging research. There is currently no National Institute of Aging like the US NIA, and the funding allocated for aging research is also pitifully small.
One thing that's very interesting, I love, as I typically do on this show, delving into our guests' other research passions. And everyone always has a few papers mixed in with their work on other themes that may be tangential to the core. In your case, you're very interested in two really interesting fields. One, nuclear fusion, which has been a very hot topic for us, and we've been spending a lot of time, again, with thought leaders in that area, both in the United States and Europe, as well as dinosaurs, which are always cool to hear about and think about. Obviously, two different areas of research that are kind of disparate from aging, but love to hear, about your other research passions while we have you today.
Humans are very interested in the question of why dinosaurs suddenly disappeared at the end of the Cretaceous period, and over 100 explanations have been proposed. However, most people believe that dinosaurs became extinct due to a violent asteroid impact. Yet, this theory has many flaws. For example, calculations indicate that the dust ejected by the impact could only produce a brief nuclear winter, even if it is revised to soot from forest fires caused by the impact. The duration of the nuclear winter would only be a short 3 to 5 years. which would not lead to long-term climate change. Moreover, the nuclear winter hypothesis also fails to explain why cold-intolerant Mesozoic relic species such as crocodiles, turtles, Komodo dragons, and tree ferns can still survive to this day. Fossil evidence also shows that after the asteroid hit the Earth, dinosaurs continued to survive for 700,000 years before becoming completely extinct. In addition, I have analyzed other theories of dinosaur extinction and believe that the likelihood of these theories causing the extinction of dinosaurs is very small. So, how did the dinosaurs become extinct? I believe that at the end of the Cretaceous period, the global temperature dropped rapidly, causing ice caps to form at the poles. Since snow and ice strongly reflect sunlight, the temperature would further decrease. This led to the fixation of atmospheric carbon dioxide in the form of organic carbon and hydrated methane in permafrost and the seabed. As a result, the concentration of carbon dioxide in the atmosphere became too low. The evidence is that the concentration of atmospheric carbon dioxide was 2,132 parts per million in the late Triassic of the Mesozoic era. In 1992, Serling's research found that by the end of the Cretaceous period, the concentration of carbon dioxide had dropped to only 229 parts per million. This was not enough to meet the needs of plants, which required high concentrations of carbon dioxide for smooth photosynthesis. As a result, plant growth was slow, and the nutritional value of forage per unit mass was low and coarse, making it difficult to digest. This led to herbivorous dinosaurs becoming extinct gradually due to food shortages and malnutrition, producing thin-shelled eggs that could not hatch. It also led to the extinction of carnivorous dinosaurs. The evidence is as follows. Evidence for the formation of ice caps. In 2016, Stanley pointed out that among the 11 major extinctions, the Precambrian, Ordovician, Devonian, Cretaceous, and Eocene extinction events all have evidence showing that continental glaciers were quite developed in the polar regions, and that there was a regression of the sea in their later stages. In the 5 major extinction events, there are residual deposits of continental glaciers. Evidence that temperature decrease leads to a decrease in atmospheric CO2 concentration. Petit et al. Found that in the glacial periods of the past 500,000 years, temperature decreased first, followed by a decrease in atmospheric CO2 concentration. In the early warm period of the Carboniferous, the CO2 concentration was over 8,000 parts per million. However, as the Earth entered the late Paleozoic Ice Age, the atmospheric CO2 concentration dropped to a minimum of around 100 parts per million. Evidence for food scarcity and dinosaur malnutrition. At the Cretaceous-Tertiary boundary, terrestrial plants became shorter and sparser compared to before, and no land animals weighing more than 25 kilograms were found. Chickens with malnutrition lay thin-shelled eggs, and many thin-shelled dinosaur egg fossils were also found at the end of the Cretaceous period. Evidence that decreased CO2 concentration led to a decline in the nutritional value of fodder. Researchers from the UK and Germany simulated the climate of the dinosaur era by raising the CO2 concentration in greenhouses from 400 parts per million to 2,000 parts per million and grew a variety of Mesozoic plants. The analysis showed that the nutritional value of the plants was much higher than previously estimated. In terms of dry matter, a 30-ton sauropod dinosaur would only need to consume 51 kilograms of plants per day, compared to plants grown at a CO2 concentration of 1,200 parts per million. It could eat more than half less. However, at the end of the Cretaceous period, the CO2 concentration had dropped to only 229 parts per million, making a mass extinction inevitable. Evidence that the fodder was coarse and hard to digest. In the late Cretaceous period, hadrosaurs evolved with over 2,000 teeth capable of grinding fodder. Interestingly, I have also discovered the most ideal geoengineering solution to control global warming from the mechanism of dinosaur extinction. You see, the global annual emission of carbon dioxide reaches over 40 billion tons, which keeps the temperature rising, gradually melting the polar ice caps, and causing permafrost and the Arctic Ocean to absorb more solar heat radiation. This, in turn, leads to the release of carbon dioxide and methane from permafrost and the seabed, further increasing the temperature. The climate will become as hot, humid, and high in carbon dioxide concentration as it was in the Mesozoic era. Many mammals will not be able to adapt to such a climate. The global ecosystem will collapse rapidly, and a mass extinction will be replayed. In the face of the annual emission of over 40 billion tons of carbon dioxide, the various carbon reduction plans and geoengineering controls that have been proposed are like a mantis trying to stop a chariot. I believe that cooling the Earth is quite simple. Install tidal power turbines in the Bering Strait. This can not only generate electricity, but also reduce the warm currents entering the Arctic Ocean, thereby expanding the sea ice area to strongly reflect sunlight, which in turn leads to a drop in temperature. As a result, permafrost and the seabed will no longer release carbon dioxide and methane, and atmospheric carbon dioxide will gradually be fixed in the form of organic carbon and hydrated methane in permafrost and the seabed. The geoengineering solution I propose to control global warming is also feasible, because historically, the Bering Strait has been closed, leading to a significant drop in temperature. Nuclear fusion is the ultimate energy source for humanity. To achieve controlled energy release from nuclear fusion, the fusion fuel must be heated to temperatures of hundreds of millions of degrees, under high pressure and high density. However, no material can withstand the extremely high temperatures of such a plasma. The current solution is to use a strong magnetic field to confine the plasma. The problem is that the strongest magnetic fields currently available can only generate pressures of 2 to 3 atmospheres. As a result, the plasma density is close to a vacuum, making it very difficult for fusion reactions to occur. Additionally, the magnetic field is very unstable, which leads to a short confinement time for the plasma, and thus an incomplete fusion reaction. Moreover, the fusion process releases high-energy neutrons, which can rapidly embrittle the first wall material of the plasma confinement device. This issue is also very difficult to resolve. The solution I propose is to have a spherical hollow container rotate both longitudinally and laterally at the same time. The centrifugal force generated by this rotation will cause liquid metal, such as lithium or lithium-lead alloy that has been pre-injected, into the spherical hollow container to uniformly line the inner wall of the container. This will isolate the high-temperature plasma in the center from the liquid and gaseous metal. The advantage of this solution is that it can confine the plasma for a long time and at high pressure and density, in addition to preventing the plasma from burning the inner wall of the reaction container, shielding neutrons and reflecting heat radiation from the plasma. The liquid metal layer also has the advantage that when neutrons hit lithium, they produce tritium, a rare fusion fuel. As shown in figures A and B, the reaction vessel 1 is equipped with rotation axes 2 and 3, which are driven by motors to rotate the reaction vessel 1, both longitudinally and laterally. The reaction vessel 1 is a spherical hollow container, The inertial centrifugal force generated by the longitudinal and lateral rotations causes the liquid metal to uniformly line the inner walls of the pressure-resistant shell 4 of the reaction vessel, forming a liquid metal layer 5. The surface of the liquid metal layer 5 is heated and vaporized by the high-temperature plasma 7, producing a gaseous metal layer 6. As a result, the plasma is confined at the center of the reaction vessel, forming a spherical ultra-high temperature plasma 7.
Bilu, what is coming up for the next part of 2025 as we get close to the end of the year? Plans for 2026 related to your research, to the company, to anything else while we have you today, please.
Since the company hasn't raised any money yet, we can't conduct in-depth research. All we can do is to publicize the TRCS model as much as possible. I hope someone will understand my aging theory and then invest in or sponsor my experimental research. Human life is too short. The life expectancy in China and the United States is only 78 to 79 years. Statistics from the Hevolution website shows that the global average life expectancy will be 72.3 years by 2050. Even in the world's longest-lived regions, such as Hong Kong, Madrid, Okinawa, and Singapore, the average life expectancy is only 85 to 86 years. It is already quite rare for people to live past 90. We need to race against time to work out effective measures to intervene in aging as soon as possible. That's all. Thank you for your listening. And once again, thanks for having me on the show, Ira.
For everybody out there that is going to be listening to this episode of our show across the various podcast networks, or who will be watching on the YouTube channel, again, you've been spending time with Bilu Huang, head of Fuzhuang Therapeutics in China. Bilu, I want to thank you for taking the time out of your schedule to talk to us for a little while today about everything you're up to. Thank you for what you're doing there in China. And as we like to say on our show, thanks for helping to create new ideas and new concepts that ultimately lead to a better tomorrow for all of us. It was really great having you on the show.